Dear friends, today we will be studying the basic elements of paleobotany, and we will try to study the plant fossils and their morphological details. The basic difference between the study of the plant fossil and the study of the animal fossil is that the animals, they get preserved with their whole body many at times, but this is not certain always with plants. It is very difficult, rather impossible, to have a situation in which the roots, stem, leaf, fruits, seeds are preserved at the same time and at the same place. Sometimes you get stem preserved without any root or leaf. Sometimes you have only leaves preserved without any evidence of the supporting stem and roots. Sometimes you can have only roots preserved without any attachment with the stem or leaves. That kind of a natural constraint of the preservation of the plant remains. It becomes very difficult to put or to assign the observable fossil any paleobotanical or biological affinity. To cater or to address such problems, the concept of form genus has came into being. The concept of form genus says that when you have any object related to the plant preserved before you, then you analyze, you study that particular morphology and compare with the similar morphology. That means the leaves will be compared with the other leaves, the stems will be compared with the other stems, and we have to accept at this point that it is really difficult or impossible to have all the features of the plant preserved at one place. With this idea or with this overview, we will try to study the plant fossils. In India and elsewhere, the plant remains are usually attached with the coal deposits or the coal seam. In India, the best plant fossil preservation is found in the Gondwana rocks, where you have the thick coal seams, thick deposits of the coal. We get plant fossils into the early or the lower Gondwana and upper or the late Gondwana. We get plant fossils in some other parts as well, like we get plant fossils of the palm in the intertrappean beds of the Deccan Traps of Cretaceous age. So, we will be studying with the morphological details in this lecture. Let us start. Here I have a rock which can be very safely said as black shale, and on this black shale you may be observing this feature. So, these are all the features which are impressions of a leaf. The age of this rock is Permo-Carboniferous, and it is classically assigned as lower Gondwana rocks, and this particular leaf is termed as Glossopteris. Now, the Glossopteris is extinct. We do not find any modern counterpart of the Glossopteris, but we know that they were pteridophytes. So, what should be observed in a fossilized leaf? We will be observing the shape and size. This is medium sized leaf. The apex of this leaf, rather than being acute, it is broad. The margin of this leaf is smooth. it has got a well defined midrib, and it has got the reticulate venation. You may have studied the parallel and reticulate venation in your early classes or in schools. We will be simply revising it here. So, in the modern leaves you would have seen reticulate venation, but now we are seeing this in a fossilized leaf. So, this Glossopteris is characterized by its oblong, almost oval leaf with broad apex, entire margin, well defined true midrib and reticulate venation. When you get such leaves with every morphology similar, but without a true midrib, then a separate genus is assigned, and that is called as Gangamopteris. So, this particular specimen is of Glossopteris. Why? Because I identify the true midrib. In Gangamopteris, the midrib is pseudo. We get pseudo midrib. What is pseudo midrib? When many of the veins they coalesce in the center to give an appearance of a midrib, but actually it is not a true midrib. So, that kind of a feature is present in Gangamopteris, and in Glossopteris we get the true midrib. So, this is a fossilized leaf of a pteridophyte. During the Paleozoic time, the earth was dominated by the pteridophytes as well as gymnosperm. This one is one kind of a leaf. We have another leaf. This is a pteridophyte leaf. Very commonly this is called as horsetail. Horsetail see a central axis, and from the central axis you can see a bundle coming out in opposite direction, and this is characterized by the veins running parallel along that. This particular form is called as Schizoneura. Schizoneura is also now extinct, and this is preserved in the black shale. The Schizoneura form is basically a form in which we get bamboo like bundle in the center, from which the leaf shoots are given in the lateral direction in both directions. Here the phyllotaxy is opposite because the leaves are given off in opposite direction. Had it been one after the other, so it would have been an alternate, but here it is opposite phyllotaxy. So, the two leaves which I have described here, they are all simple leaves, but then in the upper Gondwana we get the compound leaves also. Here I see the leaf of the Elatocladus, which is a part of the gymnosperm. So, this is a gymnosperm leaf, compound leaf, and what makes this compound is that the interdigitations on the margin, the notches in the margin of the leaf, they touch the midrib, and when they touch the midrib you get another pinna, and you get a lot of pinnas here. But if you see from the true midrib, the angle that individual pinnas subtend is almost 80 degrees. If this angle is found to be 90 degree, then this form will be termed as Ptilophyllum. So, the basic difference between the Elatocladus and Ptilophyllum is the angle that the individual pinnas make with the true midrib. If it is perpendicular, you can say that it is a Ptilophyllum, and when it makes an angle of about 70 to 75 degrees or even 80 degrees, then it is Elatocladus. So, there is not much difference. Apart from that, The other leaves which I have shown you, they were preserved in the dark colored rocks. This one is preserved in a lighter colored rock, and that means sometimes it gets preserved in ash beds or it gets preserved in certified rock. So, the matrix of preservation might vary from one specimen to the other. So, this was about the leaf. The fundamental features to be observed while studying the fossilized leaf is that whether the leaf is simple or compound, the apex is acute or broad, the margin is entire or it is with lobes or serrated, whether we do have the true midrib present or not. At many places you may get even petioles that get preserved, but in some of the specimens it is also not found preserved. After studying the morphology of the leaf, now we will be discussing the morphology of the fossilized stem. The way in which the leaves get preserved in the rocks, in the sedimentary rocks, and the way in which a stem gets preserved in the sediments, they are very different. The leaves get preserved by carbonization or compression, whereas the stems get preserved by a special process called as petrifaction, and this happens to be the best mode of the fossil preservation. What is petrifaction? Petrifaction is a process by which the individual tissues in the stem is replaced gradually by silica. Hence, if you cut a transverse or the longitudinal section of a preserved stem preserved by the process of petrifaction, then you will be able to see the individual cells and tissues exactly the way they look in a modern biological specimen. But the material which you are looking is not an organic material. The organic material of the tissues have been lost and this has been replaced by the silica. Mostly silicic acid, they replace the tissues and the entire biological structure is preserved very beautifully, and this process is called as petrifaction. Most of the stem is preserved in the stratigraphical record by the process of petrifaction. As you can see here, this is a palm wood and you can see the individual tracheids and weasels. So, and wherever they intersect, you can see some pits formed by that. So, they are very beautifully preserved by this process. No other process can preserve the individual stems like that. Then the other plant remain which is to be studied. Again, this is from the Gondwana, lower Gondwana rock, and this is popularly called as vertebraria. Vertebraria is a preserved root, and this in some specimens have been found associated with the Glossopteris leaf or Glossopteris stem. So, people believe that the vertebraria represents the root of the Glossopteris. And as you see here, you can see some central axis from which you have some transverse bars coming out. The matrix of preservation is black shale. It might get preserved in some other rock types as well. This is another specimen in which you have more than one central axis and the transverse bars. These transverse bars, they represent the rootlets. There are many more plant fossils preserved here, apart from which sometimes the fruits also get preserved, and that kind of a preservation is called as fructification. The more organic matter which is available when the entire system gets buried, the more silicic acid which is released, and it promotes or it fastens the process of fossilization. The other mode in which the plant remains can be fossilized is the presence of spores and pollens, which are too minute to be seen with the naked eyes. So, you need the aid of the microscope to study them. That means a potential unfossiliferous looking like rock can be proved to be fossiliferous if you analyze and macerate and look for the fossils under a microscope. Interestingly enough, the most of the plant fossils, the variety of the plant fossils in Indian strata is found in the Gondwana rocks, lower and upper Gondwana. And as far as the lower Gondwana rocks are concerned, so they are very close to a very great catastrophic event in the history of Earth, and that is the Permian-Triassic mass extinction. So, Permian-Triassic mass extinction, you may be knowing that more than 95% of the marine life form, they became extinct, but the plant fossils, they had a very little effect of the mass extinction. That means, the mass extinction effect is almost not felt in the stratigraphy of the Gondwana rocks, and because of which we could get some useful fossils, and that were very much helpful and significant in understanding the paleofloristics of the upper Paleozoic strata. And as far as my knowledge is concerned, the plant fossils, as far as their relationship with the sedimentary record is concerned, the rivers that existed before Devonian, that means the pre-Devonian times, they did not have meandering river, and that has very much to do with the evolution of land plants. You may be surprised, what is the connection between the meandering or the flow pattern of the river and the evolution of the land plants? The answer to this question is that if you see a meandering river, to create the meandering river, you need the stabilized bank, and bank stabilization is only possible when you have the thick vegetation on the sides or on the banks of the river. But since the land plants were not evolved before Devonian, therefore, the river system could not make itself meandering. That means, mostly the fluvial pattern in the early Paleozoic or pre-Devonian time is braided. This is only one type of the uses of the plant fossils. There are many insights which the paleobotany throws into our understanding of the evolution of the life on Earth. Till our understanding goes now, we have also used the plant remains for the hydrocarbon exploration and for many other purposes. In this lecture, I have tried to give you a very simple overview of how to study the morphological details of a fossilized leaf, fossilized stem and fossilized root. And we will be studying about the more of the paleontological groups in the lectures to come. Till then, thank you very much.